This week we're going to take a look at chromosomal material, genes, and DNA from a completely different perspective. Rather than looking at what happens inside of an individual cell, we're going to step back and look at how the protein products that are coded for by DNA can be expressed in the phenotype or the physical appearance of an organism. And we're going to understand how the alleles that you receive from your mother and father interact with each other to ultimately determine the physical appearance that you have. And this is the topic of Mendelian genetics. And if we're going to talk about Mendelian genetics, I think it would be a good place for us to start by talking about who the heck Mendel was in the first place and why we call this Mendelian genetics. Gregor Mendel was an Austrian monk. He was a contemporary of Darwin. In other words, he was alive at the time that Darwin was alive. He worked part-time as sort of like a substitute physics and math teacher at a local high school in Austria, but he was also a monk, and he spent a lot of time doing research. And his area of research was, or his area of interest was studying garden peas, the kind of peas you eat at dinner on your plate. And what he attempted to do through very, very careful crossbreeding of different types of pea plants and very careful record keeping and statistical analyses of his results of those matings, he was able to refute the blended inheritance belief that was widely accepted at that time as the way to describe how traits were inherited from parents. And the blended inheritance belief went something like this. If your mother is tall and your father is short, the blended inheritance belief said, well, you'll be halfway between those two, you'll be medium height. What Gregor Mendel was able to show, and what we're going to see very shortly here, is that in fact oftentimes with alleles, one allele will be dominant to the other. In other words, it will have the ability to cover over any influence the recessive allele might have on your phenotype. So if one person is short and the other person is tall, if the tall allele is the dominant allele, then the offspring will be tall. You won't get this sort of blending of tall and short to make medium. You're going to either be tall or you're going to be short, one or the other. Okay, so let's look at how Gregor Mendel did this. Gregor Mendel had lots and lots and lots of garden pea plants. He spent a lot of time in the garden raising these. And he spent a long time at the beginning of his research developing what he called true breeding strains. And what that means is these are strains of peas that produce the same quality or characteristic generation after generation after generation without fail. So for example, he had true breeding short pea plants. He had true breeding tall pea plants. He had true breeding pea plants that made green seeds. He had true breeding pea plants that made yellow seeds. He had ones that made only wrinkled peas. He had ones that only made smooth peas. He had ones that made only white flowers. He had ones that made only purple flowers, etc., etc. So he had all these true breeding strains. And then what he did was, when he knew he had a strain that was true breeding that would produce the same offspring generation after generation after generation, he would cross it with another different true breeding strain to see what would happen. And what he was really doing was selecting for different phenotypic traits, right? He was looking at the physical appearance of these pea plants and selecting for traits that he could see visibly with his eye, and that's different from selecting for the genotype, right? Because he didn't have any kind of technology available at the time to know what the genetic makeup was of each of these plants. In fact, people at that time didn't even know what DNA or genes were. That wouldn't come for more than 100 years before we really knew that. So he was basing all of his experimental design on phenotypic traits and crossing these true breeding plants to see what would happen in the offspring. So for example, he took a true breeding tall plant. It had two dominant alleles for having a tall height, and he crossed it with a true breeding short plant. I'm going to represent the short allele with two small t's, and he wanted to see what would happen. Now the blended inheritance belief would have predicted that all the plants out of that union would be of medium height. What what he found, what Gregor Mendel found when he crossed these two together, was that all of the offspring of the first generation, what he called the F1 generation, were all tall. Not at all what you would have predicted if you believed in the blended inheritance hypothesis. So he was able to refute right away and say the blended inheritance hypothesis is not correct. Look at what happens when I combine tall and short plants. I get tall plants, I don't get medium-sized plants. What was interesting is what he found out what happened when he combined two individuals from the F1 generation. Each of these individuals would have been big T, little t. If we combine two of them together, when he combined them together, what he found out is that he got 3 tall plants for every 1 short plant. So there was an occasional short plant that would pop up from time to time. And this led him to hypothesize that some traits are what we call dominant traits, and some traits are what we call recessive traits. And we'll explain shortly what these terms mean and how they play out in terms of phenotype. Now, one of the interesting things that Mendel found, sorry, I need to back up here. Yeah, one of the interesting things that Mendel found was something called what he called his law of segregation. And what this said was that, for example, if he were tracking the breeding of tall versus short plants, and at the same time he was also tracking in the same plants what happens when you cross the tall plants with green seeds with the short plants with yellow seeds, what kind of events do we come out of that? What he found was the traits segregated independently, meaning that the tall and short traits were passed along to the offspring independent of the traits for having green or yellow seeds. And what we know today is that's because those two traits were on different chromosomes. So what we know is that those alleles were located on different chromosomes. You all know what alleles are. The position where a gene is found on a particular chromosome is called its locus. Okay, so as long as the loci, that's plural for locus, are on different chromosomes, let's say the chromosome for height is on chromosome X1 and the chromosome for seed color is on chromosome 2, that means these can each be independently passed to offspring. There's no interaction between the two. If both seed color and height were on one chromosome together, then you would always have tall green plants, or you might always have short yellow plants, but you'd never have tall yellow or short green. Okay? This is called law of segregation. And he was able to show this through phenotype through all of his crossbreeding. Hundreds of years before we knew what a chromosome was or what meiosis was and how things segregated and how we had metaphase and anaphase occurring inside cells. Pretty amazing. Now, one of the things I told you that Mendel did was he was able to discover that some traits are dominant, other traits are recessive. I want to be very clear about something. A lot of people have a misunderstanding and think that the term dominant means the most commonly seen trait in a population. That is absolutely false. And I'll give you a great example of this. Think about in the United States, what do you think is the most commonly seen hair color in the United States? If you said brown hair, you're right. What do you think the most commonly seen color of eyes is in the United States? If you said brown eyes, you're right. Having brown hair and having brown eyes are the most frequently seen traits, but they are not necessarily dominant. Okay? If we went to Norway, okay, I'm from Norway. In Norway, or in any place in Scandinavia like Sweden, individuals there, the most common hair color is blonde hair. The most common eye color is blue eyes. Guess what? Blue eyes are recessive. Okay, and yet they're the most commonly seen type of trait in that area of the world. That just means there are more copies of the blue eye allele floating around in the population. It doesn't mean the blue eye allele is dominant in the sense of being more common, okay, in that way. So dominance means that it is what is phenotypically expressed. Dominance has nothing to do with how common it is in the population. Huntington's disease is a dominant disease, and yet only about 1 in 25,000 people in the US have Huntington's disease. It's very rare, even though it's a dominant disease. Dominance has nothing to do with frequency. Dominance has everything to do with determining what is phenotypically expressed. If being a tall pea plant is the dominant trait, then that's what will be phenotypically expressed when paired with a recessive short allele. It doesn't mean that that's the most commonly seen height of plant. It just means that anytime you have the dominant tall allele paired with any other allele, the tall allele will be the one phenotypically expressed. And what that means on the recessive side is that in order for a recessive allele to be expressed at all, it has to be paired with another recessive allele. If it's paired with a dominant allele, well then the dominant allele will be the one physically or phenotypically expressed. So let's talk about a few little bits of terminology here, and in class we'll practice with some of these so that you get it down for sure for yourself. Anytime we're referring to a dominant allele, we're going to use a capital letter. So if we were talking about plant height, I'd use capital T for tall. That would be, the fact that I use the capital letter would indicate that that allele is the dominant allele for that trait. A small case letter would be used to represent the recessive version of the allele. Notice that I use a T for both. I don't say little s for short. That gets really confusing. Use the same letter for a trait. Just use either capital or small letter. Now, if a person receives 2 copies of the dominant trait, one from its mother, one from its father in the form of egg and sperm, we say that that person is homozygous dominant. Homo means same. Zygous comes from zygote, the union of egg and sperm. That means you have the same allele from the egg and the same allele from the sperm united together, and they were both the dominant allele. Homozygous dominant. An individual who's homozygous recessive would have 2 copies of the recessive allele. A heterozygote is an individual who has 1 copy of the dominant allele and 1 copy of the recessive allele. Hetero means different. Zygote means union of egg and sperm, so you have a different allele from the egg and a different allele from the sperm uniting together. These are all here, everything that I've shown you here, those are genotypes, right? The genetic makeup. And these are representative of diploid cells, aren't they? Because they show you an allele that you got from mom and an allele you got from dad. Those are diploid genotypes. What they code for, a homozygous dominant here would have a phenotypically tall appearance. That's the phenotype. Okay, the phenotype for a homozygous recessive would be short, and the phenotype for a heterozygote would be tall, and the reason is anytime a dominant allele is present in the phenotype, it will be in the genotype, it will be the one phenotypically expressed. The only way I can get the recessive trait expressed phenotypically is if it's paired with a copy of itself. Okay, so genotype, in other words, determines the phenotype. Your genes, we know, code for the proteins that are expressed in your physical appearance. So again, if I already said this before in the previous one, but if I am homozygous dominant, I have 2 copies of the dominant T allele, I am tall. If I am a heterozygote, a big T and a little t, I'm tall. If I am this should have been 2 little t's, if I'm a homozygous recessive, then I am a short plant. Okay, guess what? On the next round we're going to start looking at some of Mendel's experiments he did, and then we're going to get a chance for you to play around with trying to solve some genetics problems.